Let's get started. Welcome everyone to High-Affinity Talks. I'm Stan Paslavsky, CEO of my laboratories, and this is our series where we bring in people doing genuinely interesting work at the frontier of antibody discovery and have a real conversation about it. So today I'm very pleased to welcome Andrew Bradbury, Chief Science Officer and co-founder of Specifica. And the topic that we are discussing today is very, very interesting. Obviously, for the last few years, just about every company in our space has told about AI and how AI is transforming antibody discovery. So that's why it's so interesting today for everyone to see whether it's real. So our guest has led and co-organized the experiment, the first properly blinded head-to-head wet lab validated benchmark for AI and antibody discovery. And I think it's the closest thing in our field to CASP moment in some sense. Before I hand over, Andrew, just 30 seconds introduction, why we're organizing this and who we are in general. So for those who don't know us, we built MyXR. If you work with immune repertoire sequencing, chances that you or some of your colleagues used MyXR as a tool are really high. And that's exactly why this topic and what Andrew built and discussing today is very close to our home, because every day we live with the question, how much real signal we can extract actually and pull from NGS selection rounds. So when we saw that someone has built the benchmark for exactly that question and answered it with real antibodies and real measurements, we knew that we should invite you, Andrew. Well, thank you very much for joining. And with this, I'm stopping to share. And please, Andrew, share your screen and the floor is yours.
Okay. Are you all seeing that?
Yes, it's good now.
Okay, perfect. All right. So it's a good time to give this talk, as the paper that describes the results I'm about to give an overview of is in press in Nature Biotech, and we haven't got the PDFs yet, but we're awfully close to being published. So this will be coming out soon. And so this is work that myself and Frank Erasmus put together. It was inspired by going to meetings and discovering that I was basically going to be retired. AI was going to kick me out of my job because AI was going to be able to do everything. And so being a bit of a skeptic, I thought, well, maybe we need to examine this. Anyway, the first thing I think it's interesting to do is to ask AI what AI thinks it can do in antibody discovery. And so this is a combination response coming from Gemini and Claude. So what does AI say it can do in antibody discovery? So it claims it can accelerate identification of antibody-antigen interactions, improving specificity. It can design novel antibodies with tailored binding properties to specific epitopes, can improve antibodies in terms of affinity, immunogenicity, stability and solubility, manufacturability, and those two last bullet points come under the concept of developability. And the use of AlphaFold and similar tools can help accurately predict 3D structure of the antibody and how it binds to its antigen, providing atomic level insights for rational design. So if all this is true, then basically AI only needs experimentalists to really just confirm what it's coming up with and test its models. So we wanted to test those claims, and because we didn't know the truth, I'm old enough now to have lived through a few hype cycles, and I'll be honest, I contributed to, I think, some of the in vitro display hype cycles earlier in the early 2000s, thinking that phage display at that time was going to take over antibody discovery. It turns out that it hasn't. But it is definitely contributing about a third, 40% of new antibody therapeutics. So because we didn't know the truth, we wanted to make it as easy as possible. And so we chose as the target the most studied protein in history, which is SARS-CoV-2 RBD. We provided data sets based on selection and outputs from in vitro libraries. And I'll talk a bit about how we make our libraries and why this is important. These have constant frameworks and diversity only in CDRs. which means that all the analysis only needs to touch the CDRs. You don't need to play around with frameworks. And we provided very deep, high-quality next-gen sequencing datasets. This is something we're doing commercially as well, so we've been working on this quite a bit. These are based on positive yeast display populations, and we also provide extensive affinity data, which is not normally present until much later in a campaign. So in that sense, whilst we were trying to understand how valuable AI could be in a real commercial antibody discovery pipeline, we did provide more data than is usually available. Antibodies coming from our library, about 80% of those antibodies are developable. This is just the way we design the library. And what tends to determine whether an antibody coming out of the library is developable is the HCDR3 clonotype. So how do we make these libraries? Well, we start with framework scaffolds, which are based on developable therapeutic antibodies. Our rationale was that clinical big pharma has taken the antibodies, they've done the best they can in terms of developability. So this provides a good track record. We also chose them on the basis of their structural diversity. This should reduce developability risk. Hang on, let me just get a pointer. Okay. The next, the diversity in LCDR1 to 3 and HCDR1 to 2 is what we call replicated natural diversity. So this comes from NGS datasets that we've developed, where so essentially every CDR in the library is a natural, it's a real CDR, it's not an artificial CDR. It is matched to the germline gene framework. We've purged all sequence liabilities, and this again we think should reduce developability risk. The HCDR3, the diversity is too high to use oligonucleotide-based synthesis. This I should mention. I mean, it's much. I'm really glossing over the details of how we make these libraries, but you can find all the details in the publications and the patents. The HCDR3, the diversity is too great, and so instead, for what we do there is we amplify the HCDR3 from 10 donors. And we sequence that to ensure that we have at least 10 to the 8 unique HCDR3s in every library we deliver. The donor set is exclusive to every partner, the idea being to avoid or reduce as much as possible the possibility of two partners selecting the same antibody against the same target. This provides high affinity, diversity, and developability of binders. And then finally, the selection platform we use is phage plus yeast. This has been developed over the years. We found this has been incredibly effective at basically being able to do complex selections rather straightforwardly. Everything is in vitro, and we can get very rapid discovery using this approach. So what do the antibodies look like? So looking at VL, so we've got all the VL frameworks in the VL are coming from therapeutic scaffolds. All the CDRs are coming from our NGS database, purged the sequence liabilities and matched the germline. For the VH, it's similar, except that it's only VH frameworks 1 to 3 that come from the therapeutic scaffolds. Human HCDR1 and 2 come from our dataset and are synthesized, and HCDR3 and framework 4 all come from the unique set of 10 human donors. We internally do selections using the single chain FV format. We also we do deliver Fab libraries. We also have VHHs, but we're not going to talk about that here. So I mentioned we use phage and yeast display. So basically, the libraries are at least 10 billion diverse clones determined by sequencing. We use 10 to the 12 phage per selection. And the real purpose of doing phage display is just to reduce that 10 to the 10 diversity to about 10 to the 6. So something we can do on yeast. And having done the selection in phage, we deliver to yeast. And that allows us to do fine-tuning of affinities, specificity, agonists, antagonists. I mean, it really allows a great deal of complexity in the selection formats. Now, under normal circumstances, we just pick random clones, and that gives us enough diversity to give the client what they need. However, in the case that we don't get enough diversity or much more diversity is required, we also do next-gen sequencing, which is something we started doing with antibody libraries over 10 years ago now. And we've developed a pipeline for the analysis of the NGS outputs. So until recently, it wasn't really possible to compare discovery platforms. People claimed their libraries were the best in the world using different targets, different formats, and there were different biological endpoints. However, that all changed with COVID-19, which I would argue was an inadvertent natural global experiment. Everybody who knew anything about antibodies tried to get antibodies against the RBD. Everybody tested in the IgG format. Everybody tended to do the same things. These were affinity and then IC50 pseudo or live virus assessments and the recognition of variants. And there are now hundreds of peer-reviewed publications describing antibodies against the RBD. Now, in the fall of 2021, Eric Roman Safire's group published a paper, the results of which are summarized here. What she did was she opened the door. She said, send me your antibodies. And 56 different labs participated. They provided 180 antibodies. These came from immune sources, naive libraries, in silico. Anything and everything was brought in. The affinities were all measured by SPR, by Cartera. And what you have here is the spread of the antibody affinities. This is 100 picomolar, so we have about 4 or 5 antibodies with sub 100 picomolar affinities and stretching all the way up to micromolar. So when we did a similar experiment, we did selections, we did next-gen sequencing, and then we tested the antibodies by Cartera in blue, and then we also measured some of them by Kinexa in red. We were able to get over 40 antibodies with affinities better than 100 picomolar straight out of the library. Record so far was 9 picomolar, which is actually found during this AI antibody competition. And this is the data, the source data for the AI antibody challenge. That's why I've gone through this to explain all this. Okay, so this is the source data. We know that this library performs extremely well against RBD. And so that's why we provided it. Everything we've done is published. References are here, but if you do a search for me and Specifica, you'll find it. It doesn't perform just with RBD. This is a bunch of other client campaigns we've done. And essentially, over 65% of single chains, 52% of VHH campaigns generate at least one sub-nanomolar binder, and about 30 and 20% are sub-100 picomolar. Straight out of the library, no affinity maturation. Where we tend to get lower affinities and worse affinities is where the selection is complex or the epitope space is really small, like peptide MHC, and you need to eliminate cross-reactive epitopes. So question we're trying to answer here is, does AI make in vitro platforms like this redundant? I think it's important to point out that when somebody gets up and starts talking about AI and how wonderful the AI system is, you don't want to compare that, or you don't want to hear a comparison based on the way that platforms performed 10 or 20 years ago. You really need to compare with the state of the art. And I consider what we're doing here the state of the art. And so what I think people should be doing is comparing their AI antibody platforms to the way that antibodies coming out of platforms like ours and others like it perform. So our key objectives were, first, what can AI do? Providing a relatively straightforward challenge. So as I said, SARS-CoV-2 RBD, most studied protein in history, and with lots of data. Where can AI be helpful in a practical antibody discovery pipeline? And are there methods or groups with clear advantages over others? And so this was loosely inspired by CASP, as mentioned earlier, which was a protein structural prediction competition. We organized this first competition ad hoc. We originally thought about getting a consortium together to try and sort of bring in a large pool of people, but we realized it was just going to take too long. We spoke to some other people who'd done something similar and said, they told us that it was going to take at least 2 or 3 years to organize a consortium. So we decided, as time is of the essence, we'd move quickly, learn what works, what doesn't for future challenges. So everything was carried out blinded. The competition was initiated by a publication in Nature Biotech the competition was initiated and published in Nature Biotech in Q4 of 2024, and we initiated in Q1 of 2025. The NGS datasets that I'm going to describe are available for downloading by participants simultaneously, and they had 14 days to upload designed and identified antibody sequences. Now, why did we only give 14 days? The reason is we wanted to see what AI could do without any experimental confirmation or modification. The next competition will be different, as I'll explain later. The results publication is now in press. All participants are co-authors. The results are published blinded, and we thought this was important because we wanted to encourage as much participation as possible. And we understand if your business is built on AI and your algorithms do not perform, that may affect your business. And so we wanted to essentially protect those people that were kind enough to participate from the embarrassment of being demonstrated that perhaps they might be lost or something like that. However, Nature Biotech insisted that we unblind the winners, and one other participant agreed to be unblinded for a scientific reason. So every participant knows their rank within the spread, but they're not identified. This was the announcement manuscript. So we had 29 participants. We had 511 submissions altogether. You see, most were non-profit academic groups. We had a lot of AI biotech companies, and then equal numbers of small, mid-sized biotech and large pharma. We had one large, big tech company. These are the number of antibodies submitted for each individual challenge. So this is the affinity maturation challenge I'll talk about. This is affinity prediction challenge from a clustered output, and this is de novo design based on sequence outputs. This is the full data set, over 1,500 analyses. Each antibody was tested 3 times. This is the affinity maturation here. This is the de novo design here, and this is prediction of antibodies coming out of the library. One thing you'll notice is the gray ones, which tend to be SPR plots of antibodies that don't work terribly well, are more common in challenge 3, challenge 1 than in challenge 2. So how did we rank the candidates for these challenges? So first, and I'll talk about exactly how we were able to do this later, but Azenta, all the submitted sequences were sent to Azenta. They produced the submitted antibodies. They purified them and then shipped to Cartera for SPR and to Mosaic for developability. Cartera then rank ordered those antibodies by SPR and by developability. And those antibodies that were developable, including those on the borderline, were then considered for Kinexa. And I'm just showing down here the affinity range that can be tested using SPR, the LSA in particular, and BLI. Basically, it peters out at about 100 picomolar. You can get affinities less than that, but they're not as reliable as Kinexa, which can go down to the femtomolar range. So Kinexa, this was Thapidine, carried out single point screening to confirm the rank order to the SPR. And then for the best antibodies, they carried out standard screening on up to 5 candidates for each challenge, where the developability was less than 3 or less. Now, what about the developability? How did we score that? So Mosaic had 5 different challenges. So it's HIC, retention time, this BVP scores is polyreactivity, AC signs, that's self-interaction, TM, melting temperature, and TAG is the temperature at which aggregation occurs. And Mosaic has a lot of experience in this. So what they have is when they're asked to do, they have a passing threshold, if you like, a failing threshold, and then in between these two is a questionable threshold. And so what we did was we gave antibodies that passed a score of 0, those that failed a score of 2, and then those that were questionable, score of 1. And all 5 assays were added together, and in order to pass, antibodies had to get a score of 3 or less, and if they got a score of 4 or more, then they were failed. Okay, so how did SPR and Kinexa correlate? So this is a plot of all the antibody affinities measured using SPR LSA on the x-axis, Kinexa on the y-axis. This is standard Kinexa, so there's not as many antibodies here. And what you can see is there's a strong rank order correlation between standard Kinexa and LSR, but there's a poor absolute correlation, particularly at higher affinities. you see down here. And generally, Kinexta gives higher affinity values, as I mentioned earlier. That's because it's more capable of measuring high affinity antibodies. And so we've prioritized Kinexta values for high affinity antibodies. Now, if you look at single point Kinexta, we did many more single point antibodies, or Sapidine did. You see, again, we're getting very strong rank order correlation. The Kinexta screen and the LSR kinetics showed a poor absolute correlation, again, with the Kinexta tending to provide higher affinities than the SPR. The Kinexta single point screen and standard Kinexta are actually extremely well correlated, both rank order and also absolutely. And when we published this a couple of years ago, this represents a new way of using the Kinexta. I mean, it has been done, but I think we were the first to publish it with Sapidine, demonstrating that single point Kinexta test is actually quite valuable and much higher throughput. So what are the challenges? So challenge one is an in silico affinity maturation challenge. It's based on the way that we do affinity maturation in Specifica. So if this is a parental antibody here at the top, we've got all the CDRs marked in dark blue. What we do is we will take that antibody, this is the VL here, we will make an L1, L2 library, and what we do is we use the wild type plus all the CDRs in our data set that correspond to that germline gene family, purged of sequence liabilities. So first library is L1 and L2, second library is L3, third library is H1 and H2. We use H3. We don't touch H3 usually, although when we're asked, we can also do scanning mutagenesis and things like that. So then, having sorted the top clones out of each of these libraries, we then combine them into a combo library. And what we guarantee an improvement of affinity of about 10-fold, and I think our best is about 50,000-fold. The developability of the antibody tends to improve. And so the question we were asking was, okay, given these three sequence outputs, NGS, design an antibody that uses this information to provide a higher affinity. So this is what the results look like. Affinity is measured by SPR here. And so what you have in red there is the parental antibody, which had an affinity of 200 nanomolar. Those higher up on the y-axis is on-rate improvements. To the left on the x-axis is off-rate improvements. You can see that the experimental approach, which is all these blue crosses here, every single one of those, 100% of those clones all showed at least a 10-fold improvement. But this takes us an extra 2 to 3 weeks to build the library and select from it. About 30% of the AI submissions were either non-binders or extremely weak binders. This was the best experimental, and the top AI, there were 2 AI antibodies from a single organization, which improved on the basis of SPR. They improved over the best experimental that we were able to get, and they were also developable. So when we looked at these, this is taking that same data and turning it around. So what you have is now experimental here. This is the span. This is the best experimental represented by this blue line. The parental is represented by the red line at the top. And then each on the y-axis or the columns are different organizations ranked by the affinity of the best antibody they were able to generate, and showing the spread of all the antibodies they produced using their algorithms. And so you see that this AI biotech company was able to, on the basis of SPR affinity, was able to get an antibody that had an affinity that was slightly better than the best experimental, indicated here. But it also had a bunch of other antibodies, most of which I would add, I should say, were better than the parental. But then if you look down here, you see that as you go further down here, you see that quite a few organizations were able to generate antibodies that had worse affinities. And as you saw, 30% didn't bind at all. So then what we did, we took the top ranked. I mentioned we ranked those and did that by Connexa. So this is the best experimental. This is the best AI designed antibody. And then this was the second best. And what you see here is that the best AI predicted antibody was statistically as good as the best experimental. And most AI predictions were not as good as the best experimentals. So that's challenge one. Now, how much change did these people make to their antibodies? And so here what we've done is we've taken these CDRs, we've concatenated them and compared them to either the parental or the best experimental. So in dark purple, we have against the parental, and in light purple, we have against the, dark purple parental, light purple is best experimental. And you can see that actually most of these organizations had significant differences to either the parental or the best experimental. As they get worse, they tend to be closer. Then here is individual CDRs and how they compare to the consensus sequence. And the reason we did that is because one participant just submitted a consensus sequence antibody, and they were actually rather good. I think they came 4th or 5th overall. And so this is how all these other ones compared to a consensus approach, which is obviously not AI, it's merely statistical. Okay, that was challenge one. Challenge two. So we provide 3 HCDR3 antibody clusters. We've got 27F, 28F, and 47F. We provided the affinities of some antibodies in those clusters. And then the question or the challenge was, using AI or any other method, identify the highest affinity developable antibody in each HCDR3 cluster. So we provided 2,500 different unique sequences for the top cluster, 3,500 for the middle, and 400 for the bottom cluster. And these antibody clusters had different developabilities. And this is based on the HCDR3 sequence. And so this 47F cluster, pretty much all the antibodies coming out were developable. 28F, much less developable, and 27F somewhere in between. So now, although we did 3 clusters, we described 3 clusters in the publication. I'm just going to talk about one cluster. The results were similar for all. And so what you have here is you have the most abundant clone. So we've published and shown that if you do NGS, you cluster the antibodies based on the HCDR3 sequence, and you then take the most abundant sequence in terms of concatenated CDRs, that will always be a binder. However, it will not be the best binder. Usually, if you just pick randomly from the sequences, you'll find that about 30% are better than the most abundant binder, okay? And so here, this represents a bunch of randomly picked experimental sequences. This is the one of those randomly picked ones that had the highest affinity. These are, again, each individual organization in a column represented by the sort of organization they were. Where a antibody affinity, and this is measured by SPR, is developable, it's an open circle. Where it's a purple circle, that means it was not developable. And so what you see is that some of these algorithms were able to predict antibodies that had affinities that were better than the most abundant, but then that also many of the antibodies that were picked were actually worse than the most abundant. So going into one of these this challenge 27F in greater detail. So this is top ranked by Connexa. So this is the best experimental measured by Connexa. This is the best. This is the best prediction by the best organization, which was a non-profit academic for the best antibody. Bear in mind that this is just one antibody out of many that they predicted, and that every antibody that all these different organizations predicted were in the library, okay? And so what you see is quite a few were coming up with affinities that were better than the most abundant. Now, if we look at the developability, What you see here, this was 27F, this was the intermediate one. What you see is that there's a developability gap. It didn't appear that people were particularly good at predicting developable antibodies. For 28F, it was even worse. 47F, as I mentioned, all the antibodies there were developable. There was no developability gap. So the best predicted antibody had an affinity of 9 picomolar. For this cluster, 75 picomolar for this cluster, 50 picomolar for this cluster. And so these, as I said, this is an AI prediction. This is not an AI generative competition. So people are just trying to predict what's best in the library that we have already. So as I mentioned, developability was very much dependent on the HCDR3 cluster. And now, the question that's really important as to whether this is valuable or not is, how transferable are AI algorithms from one cluster to another? And are AI picks better than random picks? Okay. And so looking at cluster 27F, we actually had 2 participants that came, they were the 2 antibodies they picked were statistically indistinguishable. So they came top for 27F. How did they perform in the other clusters? Well, you can see that they came 15th and 16th ranked here. This is SPR affinities. And in this case, I think this one was disqualified, or they didn't participate. No, they used a different algorithm for this cluster. And this other one that came top here came 9th. So overall, if you look at their algorithms, 22% and 25% were better than the controls, the most abundant one. Okay, let's go to the next one. So here, whereas here we had 2 participants predicted different antibodies that were the highest. Here we had a single antibody, predicted affinity was 100 picomolar. How did they perform in other, how did they perform in other, how did they perform in, oh, I don't know what's happened, sorry about that. Okay, how do they perform in other clusters? You see this one that came top in 28F, it came 6th here and 16th here. The last one, we had a single organization that was able to pick the top clusters in the top highest affinity antibodies in 2 clusters, 28F and 47F. Affinities here, 106 and 50. But then how do they perform in the other cluster was 27F. Now, one of the competitors, the winner of Challenge 1, which was the affinity maturation, they also participated here. Some of their antibodies were disqualified because they had sequence violations. They made changes in in other words, they produced sequences that weren't found in the data set. But in the example where they were able to pull out an antibody that was in the data set, they came 32nd with an affinity of 4.7 picomolar, not micromolar. Now, if you look across at the percentage of antibodies that were picked by these different algorithms and compare them to the affinity of the most abundant antibody and ask the question, how many of those were better than the most abundant? Then, with the exception of this one, which was able to get 50%, all the others came in between 11 and 25%. You compare that to just picking clones at random, we got 39%. So in other words, what this means is that when picking additional clones at random in this data set, 39% are better than the most abundant experimental controls. And except for one algorithm, which was able to pick 50% antibodies better than the controls, the other winners all picked antibodies with better affinities, worse than if you picked random. So AI is actually working directly against you. The performance of winning algorithms also, with the exception of the winner for clusters 28F and 47F, did not really transfer across to other clusters. So this is a bit worrying because I had thought that this might be the easiest challenge of all, and it turns out that it wasn't. Okay, challenge 3. So here what we did was we provided participants with the full selection output. So we have 30,000 sequences. These are all positive by they're all positive by yeast display. We also provided the affinities of over 140 antibodies. And so the challenge was taking this sequence data set, design high affinity antibodies modifying only the CDRs that were not found in this data set. So leave the frameworks untouched. Okay, so again, this is the results all ranked by SPR. So here we have the experimental. This is the best experimental. These again are the different organizations. Antibodies that are purple are non-developable. Those that are circles are developable. And you see there's one antibody here designed by one group, the small to mid biotech, that had an affinity that was better than the best experimental. However, all of these were really too close to call by SPR. Their affinities were 20 to 40 picomolar. And so we measured Kinexa, and the affinity of that antibody that appeared to be better is actually significantly better by Kinexa. It has an affinity. This has an affinity of just under 3 picomolar. However, I'll say a bit more about this. This antibody did not elute from the Hick column, and because of the developability criteria we'd established at the beginning, it did win, but it did have a few other issues, and so I'm not sure whether this would have been a good clinical candidate. If you exclude this guy, then the next best AI-designed antibody was statistically indistinguishable from the best experimental, which is about 9 picomolar. So there were different algorithm classes, as indicated here. So statistical heuristic, that was the consensus sequence analysis, which did quite well, as I said, in the affinity maturation, but less well elsewhere. There was classical machine learning, PLM attention-based, structure-aware, and then there was one that was undisclosed. This is the number of participants using each of these different approaches. If you compare these across the different challenges, there's challenge 1, challenge 2, the 3 clusters, and challenge 3, the AI, the out of NGS dataset design. What you see as the different algorithms perform differently, but there is no one algorithm that is consistently the best across all 3 challenges. And the other thing is that if you look at the best algorithms for each one and see how they compare across the challenges, so this is now classifying the AI approaches by their algorithm class as opposed to by the organization. So here we have the top in challenge 2, prediction challenge. You see that when they in challenge 1, which is affinity maturation, they came 11th. So this sorry, I should explain. The rank here is post disqualification for the first number, and the second number is pre-disqualification. And disqualifying is either because the sequence was wrong or because the antibodies were non-developable. And so if we look at the first number, you see this PLM attention came first for this particular cluster, then came 6th, 16th for the other 2 clusters, and 11th for affinity maturation. It did also, however, come first in challenge 3, which was the design. Looking at this one, came top for this cluster, 15th for this cluster, 20th for that cluster, 4th for challenge 3, and 105th in the affinity maturation. And so you can see all the way across, there is no cluster, there's no challenge, there's no method really that is uniformly successful across all of the different challenges. And so conclusions, much more depth in the paper, which, as I said, will be coming out soon. I believe that with high quality data sets, where you have extensive affinity and next-gen sequencing information, I think for challenge 1, so I think you can generate higher affinity antibodies. Now, the question is this approach consistent? If it was, it would save 2 to 3 weeks over our experimental approach, which we know works. And so we're now thinking of carrying out in parallel, or we will be carrying out in parallel, AI type approaches in parallel with our standard experimental approach to see if it works in the real world. Identifying the highest affinity antibodies in 3 different clusters. Unfortunately, prediction was worse than random picking for all but one algorithm. The algorithms were very poor predictors of developable antibodies. Basically, the ratio of developable antibodies to non-developable was the same as the ratio within the cluster. And no algorithm was able to consistently predict the best affinity antibodies in each cluster, although one algorithm predicted best affinities in a couple of the clusters. Challenge 3, de novo antibody design, given over 30,000 sequences and almost 150 affinities. The best AI is extremely high affinity, 3 times better than the best experimental, 9 picomolar. But as I mentioned, it was unable to elute from the hit column. It had a low specific activity, and it had a cooperative binding profile, so we're not quite sure what that meant. And we wonder, but certainly this would not be clinically developable without additional work. And if we exclude that antibody, then the next best AI antibody is indistinguishable from the best experimental. Few companies showed improved distributions over experimental, although many were able to match the distributions of experimental outputs. As I mentioned, the data set, all measurements and sequences and source data is going to released to the community after or at the same time as the publication, and this allows you, if you have an interest in trying to compete against the other institutions that participated here, to see how your algorithms work. So I mentioned a few key objectives at the beginning. So understand what is really possible. Is it hype or reality? I'd say it is reality in affinity maturation, given good enough data and a good algorithm. The good results, I think, do depend upon high quality data sets. I don't believe we had an AlphaFold moment, which was in CASP, where suddenly, I think two CASPs ago, AlphaFold revealed just how powerful it was. So there was no definitive winner across all challenges, although some were not bad in terms of consistency. Where can AI be valuable? I think, as I said, affinity maturation, discovery prediction, a discovery of good antibodies out of clusters was worse than random, except one algorithm. And out of library design, I think, is valuable to expand the number of leads, but at least at the moment, it's not clear that it increases the affinity, except for in that one case of the antibody that had a few problems. Is there one algorithm or one group that performed consistently better than the rest? There were a couple that did quite well. One biotech fared pretty well across challenges 2 and 3, and there was one winner for challenges 1 and 3. And so, next challenge, what we thought we'd do is to have a bake-off between experimental and AI. And so, Frank Erasmus and I are handing over to the Antibody Society. We actually wanted to participate in this. We did get some antibodies, but we weren't including them in the results. We felt that was going to be too much of a conflict. We will like to participate in the next one, and so the target will be announced by the Antibody Society. The antibodies will be judged on affinity and developability, and in order to allow immunization approaches as well as in vitro approaches, we're going to allow 14 to 16 weeks to deliver sequences of 3 top antibodies derived using any method. So this can be in vivo, it can be in vitro, or it can use AI, including design, build, test, learn cycles. And I think this is really important to understand how well AI works in a real case scenario, in the real world, as opposed to in the world of white papers. The other thing I'd like to say is, I think when people publish their white papers, I'd encourage them to publish in peer-reviewed papers. And also, I would like to see the same sort of data depth we've demonstrated here in the AI Antibody Challenge. So with that, I'd like to thank our partners. So Specifica, which was acquired by IQVIA 3.5 years ago, provided the data sets. That was all done with Frank Erasmus. He's really been instrumental in making this work. Azenta did antibody production, expression, SEC characterization. Crystal Richardson and her team were really important there. Cartera, Dan Bedinger was responsible for doing SPR affinity characterization of those first over 500 antibodies. Sapidine, Liz Hopkins and her team did connector analyses, both single measurement as well as the standard. Biotechny, Raymond Chen at that time, he's moved on since then, provided the target, and developability assessments were done by Mosaic, and in particular, Crystalline Graph. And so with that, I'll say thank you, and I'll take any questions.
Thank you very much, Andrew. This is a great presentation. It's super interesting. We already have a couple of questions, but meanwhile, just to announce, anyone can ask question in the QA section or write in the chat. I will start with one question for myself. Yeah, it's interesting that you mentioned that the AI can help improve the affinity, but at the same time, simultaneously, I didn't get fully how it's correlated with the developability. Can it simultaneously produce a higher affinity candidate, keeping the developability good?
So I would say there's a bit of a bias here, because the way we do affinity maturation, the diversity we put in is all developable. Okay, so unlike other people who do random mutations, what we do is we use defined CDRs for every CDR. The CDR is matched to the framework of the antibody to improve, and all sequence liabilities are eliminated. So the diversity up front is developable, and we find that we almost always get improved developability with antibodies. And so, given that that's the input data and the source data, all the antibodies in those 3 NGS datasets will have generally improved developability over the starting antibody, and so that data is providing developable sequences. And so therefore, AI using that as the NGS data will be starting, if you like, on a better framework. Now, if you look at the second challenge, which is the cluster challenge, so predict the highest affinity antibody in a cluster, and if possible, improve developability, the fact that the ratio of the predicted antibodies was, in terms of developable to non-developable, was the same as the ratio of the randomly picked antibodies, developable, non-developable, suggests that the algorithms were unable or not terribly good at predicting developability.
Yeah, well, it's kind of, so for me, the question, like, you either have an algorithm that can predict for better affinity or developability, and combining this turned out to be like really hard.
Yeah, I think
kind
that's
of
fair enough.
multi-objective. I don't want to eat all the time. Let's go to questions from the audience. We have a lot. So, well, the first question is, what is your opinion on the recently published germinal method for de novo design of antibodies? Their model considers developability right from the start.
So I'm not sure I've read that paper. I would encourage the participants, the authors of that paper, to participate in the next competition to see how they really compare. Because I think I think we need benchmarks to understand. I don't think that you can really understand how well a model works just from relying on the author-based data. I'm sorry, I'm a bit cynical there.
Yeah, and a related question. So there is already a next challenge is coming, but there is a question whether you want to repeat the same challenge, later, to see whether there is a progress with the AI-based approaches. Different challenge will introduce, different questions and different answers.
So let me put it this way. I'm very happy to let somebody else do it. It was an incredible amount of work. I think it would be very valuable to repeat this challenge. I think that's a really good idea. I can mention that to the Antibody Society. Randy at the Antibody Society is taking this on now, but that's a good idea.
Next question. Were developability properties explicitly part of the AI predictions for any organization that participated in the study?
So I don't know that. I think they were. I can say that predicting developability was a requirement for the competition. And so everybody participating was supposed to take developability into account.
Well, and the fact that you had a very good library, that helped a lot.
Yeah.
Yeah. Did you run the interaction affinity analysis in both directions or just one? Could some of these AI misses be related to poor data with unclear binder kinetics?
All right. So given that, the SPI was only done in one direction. However, given that it was done by Cartera in their in-house, I don't think, and we've provided all the SPR plots and all the primary data is in the paper. So I think the data is good. It is possible that if you were to reverse the orientation, you would get different results. However, that would have been a bit more of a challenge than the way we did it at the moment. The Connexa results, again, they were done by Sapidine. They invented the technology, so I'm sure that their results were also good as well. So I'm confident in the quality of the data, in the way that the data was carried out as described.
Thank you. The next question is, can you comment on the value of antibodies that are high affinity and developable, but not functional? Point I'm trying to make here is why not look into function too?
Well, yeah, so I think this is a really good question. Personally, I don't think we understand function deeply enough to be able to design antibodies for function. I would argue, and this is why I think people should participate in these competitions. I think if you have an algorithm to design for high affinity, I think if I was doing it, I'd want to know how my algorithm compared to others. Designing for affinity is already, I would argue, difficult enough. Designing for function is even more difficult. And where there are some AI companies that have designed antibodies and then screened for function and found functionality, it's important to note they did not design for functionality, they designed for binding. And functionality was a very nice side effect of that binding. Is that clear?
For me, yes. So I hope yes. If you have a follow-up question, please ask in the chat. Next question: any particular VH or VL gene family performed better in AI design models?
Okay, so it's important to note that here our libraries are based on 4 limited scaffolds. And in fact, most of the data, most of the antibodies that we selected against SARS-CoV-2 came out from one of those scaffolds. I can't remember, I think it's a VH3 or a VH1, I can't remember. And then the design challenge was based on modifying only the CDRs and not the framework. So there was an inherent bias against, I mean, you couldn't use different frameworks. If you used different frameworks, you would have been disqualified. So this was a CDR, so I can't really answer that question. I can tell you slightly different, I can answer a different question, which is, do all frameworks perform equally well when you make in vitro libraries in the way we've done? And the answer to that is not at all. We have tested something like, I think, 12 different frameworks, VH and VL, and the 4 that we provide are by far the best. We've made VH2 libraries, VH5 libraries, and we just get hardly any binders out of those. We don't know why, and we suspect that the only reason they remain in the immune system is probably because they recognize some weird pathogen, which is, after all, what antibodies are supposed to do in the real world.
Thank you, Andrew. Next question is, can you comment on how good was the fold change improvement for affinity with single point mutation versus multi-point mutation for design antibodies?
No, sorry, I haven't looked at that. And in fact, but if you remember that slide I showed, pretty much all the top antibody designs of the affinity maturation had significant number of modifications. I don't think there was any single one that had a single mutation.
Yeah. Next question is, can you tell the winner, the number one for the affinity, what type, without revealing the name, what type of the AI algorithm was used there?
Oh, gosh. It's in the paper. I'm afraid I've forgotten. Hang on, let me just see if I can find it. think so. It was a PLM attention structure aware. So I'm not an AI person. So forgive me for not being up to date with everything. I understand antibodies. I'm trying to understand AI. So it's a PLM attention structure aware is what I've got here as a method.
Yeah, well, thank you. In general, related question, how did the results of this AI antibody competition perform against your expectations? And what are your thoughts on the current state, near future, and not near future on the AI and its applications in antibody discovery?
Right. So I was encouraged to see the best affinity maturation algorithms were as good as experimental. I was very disappointed that there were no algorithms able to predict, given a cluster of 3,500 antibodies, find improved antibodies. I was disappointed the algorithms were worse than random, with one exception. That really surprised me. The designs, I think, I was comforted by that. My overall takeaway from this was that if you have very, very deep data, and that includes NGS and affinities, then I think AI can be really valuable. My sense is that it's going to be very valuable in the particular data set that you're using. Now, this was the state of the art a year and a half ago, a year and 3 months ago, 15 months ago, for the 29 organizations that participated. I think that some people will say, and rightly so, well, we've moved on along since then. Certainly it appears from what I've read and from what I hear at conferences, it appears that's the case. I would also say that if your algorithms are so good, and if you are an investor that is invested in a company that is producing algorithms, I would want to know how good my algorithms are compared to everybody else and against the experimental platforms that exist now that are top of the sort of at the cutting edge. I think that in time, eventually, the way I see AI coming in, I think it's going to be a bit like the way in vitro platforms. Now, at the moment, you have basically a third of therapeutic antibodies are coming from wild-type animal mouse immunization and humanization. A third are coming from transgenics, and about a third are coming from in vitro platforms. I suspect AI will be another way of generating antibodies. This is a solved problem, generating antibodies that bind to targets. Functionality is more challenging. I think AI will be, maybe it'll all go down to a quarter. And AI will now perform a new function of generating new antibodies. I'm a bit skeptical as to whether AI is going to be able to design antibodies with function unless you have a good NGS data set of antibodies with functionality. I think in the absence of that, I think it's going to be challenging. Eventually, I think AI will form part of every antibody discovery pipeline.
As a part of our life in general.
Yeah.
Related question, a quick one. So since we don't know the participants, can you just tell that the set of participants is representative?
So I would say the set of participants was representative in terms of the academic groups and also big pharma. Big pharma, quite a few big pharma participated. I was disappointed that the noisiest AI companies did not participate.
I see.
If I was investing in those AI companies, I'd want to see how they performed.
Yeah, that's for sure. That's for sure. That's why I think one of the reasons they did not participate.
Now, well, I don't know. That's not true. I think there are other reasons why they might not want to participate. They may not want to disclose their algorithms, which I think is fair enough. And the other reason is because this was run by a company. Specifica is a company, and so that's why we're handing over to the Antibody Society for the next competition, because we want it to be truly blinded, informatically blinded. It wasn't this time. So and certainly some of the people I've spoken to from some of these AI companies have said that if it's run by the Antibody Society and informatically blinded, they would be more willing to participate.
Fair enough. Fair enough. With this, we are running out of time. So just thank you very much, Andrew. Thank you for participating in this High-Affinity Talks. Great conversation and a lot of questions, a lot of people. And thank you, Specifica, and everyone who joined. If today conversation and ideas about specifically NGS analysis sparkled some interest, that's exactly what we are doing. We are building an open source and white box software methods for NGS analysis and selection rounds. So if you're interested, you can check platform bio. And our next High-Affinity Talk will be on August 26th. Our guest will be Wyatt McDonald from Infimmune. And we will be switching from synthetic libraries to in vivo, mining human repertoires and discovering fully human antibodies in vivo. So thank you very much, Andrew. Thank you very much, everyone who joined.